Good afternoon. So today I'm going to talk a little bit different thing than the vaccine, but it is also related to potentially for vaccine therapy. As all of you virologists, you know that many viruses target different sites in the cells. Many viruses are the RNA viruses, and also the pox viruses having double-stranded DNA targets the cytoplasm for the replication. And many DNA viruses goes into the nucleus. And retroviruses, of course, integrate, they replicate there, and the influenza virus targets the nucleus where the genome replicates. So the host innate immune response is the first line of defense, is to recognize the viral pathogens. And so the broad category, the first line of defense is the pattern recognition receptors, which can recognize the pathogen-associated molecular pattern or the damage-associated molecular patterns. The immune response that elicited is the broad category. The first one is the type 1 alpha and beta interferons. They induce the antiviral state. The one that I'm going to be focusing today is the pro-inflammatory IL-1-beta and IL-18 and the cytokines, and which cause inflammation and fever and anorexia. And also the innate immune response induces the chemokines, which can recruit neutrophils, monocyte, NK cells. So related to the inflammation, inflammasomes, as I'm going to talk about, these are the inflammasomes I'm going to describe now. They recognize the foreign DNA, RNA, and proteins and damage, and they can induce the variety of cytokines, IL-1-beta, 18, and 33. Because they are potent response, they are controlled very tightly by the host cells via two pathways. And one is the signal one, which is the pattern recognition receptor, recognize the pathogens, then induces the cytokine production via the transcription factors, and they produce the inactive form of the proteins, IL-1-beta, 18, and 33. In order for this inactive form to become active, they have to be cleaved by the caspase-1. The caspase-1 itself is present in an inactive form known as procaspase-1. This procaspase has to be cleaved in a molecular platform which is known as the inflammasome. The molecular platform comprises of a second sensor of the potent PRR, which recognizes the pathogens, and that sensor, once it recognizes it, interacts with an adapted molecules known as the ASC, that in turn interact with the procaspase, and this molecular platform now activates the caspase-1, which in turn cleaves the proform of IL-1-beta and IL-18. If you look at the sensors, there are a number of second signal sensors in the cytoplasm. The number of them are there, known as the NLR families or human 200 proteins. The NLR families in the cytoplasm can recognize bacteria, DNA and RNA, viruses or crystals, antibody, uric acid, potassium influx, and anything. And in 200 protein, there are 4 of them. Majority of them are nucleus over in the cytoplasm. The well-characterized the AIM2 molecule, which can recognize anything in the cytoplasm, the transfected DNA or the double-stranded DNA that entering the cytoplasm infected cells. This is regarding any time you try to do gene therapy, the DNA entering the cytoplasm, it can activate the inflammasome activation. So, in summary of this one there, the interferon response, there are a lot of sensors in the cytoplasm which can recognize the DNA, which can activate a STING pathway and activate the TPK1 and IRF3, which enters the nucleus, which activates the number of cytokines. And the inflammasome response is AIM2 can recognize DNA and induce the inflammatory response. So when we started the project, we didn't know that. Does it anything that is the innate sensing, does it occurs for the nuclear replicating viruses in the nucleus, and does it activate both the inflammasome and interferon response? So why are we interested in the inflammation and inflammatory response? We work on Kaposi sarcoma-associated herpes virus, also known as KSHV or HSV8. It is involved in the number of malignancies, which are actually inflammation-associated malignancies, like Kaposi sarcoma, which are proliferating endothelial cells, and which is present with the virus is present in a latent state, There are a lot of inflammatory cells, and the inflammatory cells, some of them produce virus particles. The entire microenvironment is bathed with the inflammatory cytokines, angiogenic factors, and growth factors, which are actually the driving force of the case Kaposi lesions. The virus is also associated with the primary effusion lymphomas, which is also inflammation-associated cancer. And the cells can be established in culture, which can grow and continuously like EBV positive cells. There are more than 80 copy number of the viral genome is there. We can induce the virus particle from these cells, and which comes to serve as the source of the virus particles. So when we started doing this particular studies, then we said we took the virus particle, which are labeled with the viral genome, is labeled with the BRDU gene, BRDU, and you can see the green particles are all represent the virus particle. The virus enters the target cells very rapidly. Within 30 minutes, the viral DNA gets into the nucleus. Once you enter the nucleus, it is a profound inflammatory response is induced. We took the supernatant of the infected cells. These are from endothelial cells. We measured the cytokines by ELISA, and we found a number of growth factors and cytokines are produced. Among them, IFN-β is there. And this growth factor and cytokines basically resembles what is seen in the in vivo conditions. Since we found IFN-β, we asked the question of does the caspase-1 cleavage occurs? What you see in the slides, maybe from the back we're not able to see, compared to the uninfected cell, during the time of infection, increased the cleavage of the caspase-1 activity was seen during KSHV infection. So next we asked the question is which one of the sensors may be involved in it. Since the adapter molecule is the one which is mediating, interacting with the sensors, we immunoprecipitated ASC and then look for the different adapter. The cytosolic adapter AM2 was associated only at 2 hours, but only the IFN-16, which is present in the nucleus, is interacted with the ASC throughout the time of infection. If you start looking at the immunofluorescent data, at the 2-hour infection, both the IFN-16 in the nucleus and in the cytoplasm, in the ASC in the nucleus, come together and they start migrating during the course of infection to the cytoplasm infected cell. Whereas the vaccinia, which replicate the cytoplasm, you don't find anything like that. So what is IFN-16? It is a 80-colored alternate protein. It is present in all the eukaryotic cells and almost all the cells that are replicating the herpes viruses, which is this is induced by gamma interferon more specifically, and it's a DNA binding protein. It can interact with the RB and P53. It is a cell cycle arrest protein. It is a transcription modulator, but we don't know the exact functions of it. The crystallography suggests that it can interact with the the backbone of the DNA. There is no specific sequence-specific recognition. It recognizes basically a phosphate backbone. So then we asked the question of, during a primary infection of KSHV, we found the inflammasome induction. So we tried to see whether does it occurs during the latency also. This is the latently infected B cells, latently infected endothelial cells. Only in KSHV positive cells we found IFN-16 and ASC co-precipitated, and IFN-16 precipitated with the caspase-1. And similar with none of the other molecules were precipitated. These are all in vitro system cell culture, so we want to find out does it occur also in vivo. We took the normal lung patients by samples, also solid primary effusion lymphoma cells. We strained for both ASC and IFN-16. We found that ASC and IFN-16 co-localize both in the nucleus and in the cytoplasm, suggesting inflammasome activation occurs also in the nucleus in the patients. So since it is KSHV, we started trying to look at does it present in the other viral infected cells. We took an EBV, which is Epstein-Barr virus, involved in the different type of cancer. We took the B cells latently infected or the epithelial cells latently infected. We found the cleavage of a caspase and constitutively activated in the EBV positive cells. Then in those cells also, when we immunoprecipitate with the ASC or caspase-1, only IFN-16 comes together in these EBV positive cells. So we've tried to locate what is being recognized. What we found was that this is IFN-16 in the nucleus or in the cytoplasm within 24 hours. This is the viral genome labeled with the FISH probe. You can see the 3 virus particles is associated with the IFN-16. In the latently infected cells, which has more than 80 copy number of genome, all of the viral genome is co-localized with IFS16. Similarly, in the EBV positive cells, EBV genome is co-localized with IFS16. Then we looked at the simplex 1, which is the type 1 alpha herpes viruses, which replicate in the lytic cycle. We found that when you infect the cells with the genome labeled virus 1, within 30 minutes, the genome entered the nucleus, and the IFS16 in the nucleus co-localized with the viral genome. Then what we found was within 2 to 4 hours, the genome, IFS16 also comes to the cytoplasm, but 8 and 24-hour time point, the virus is completely destroyed the IFS16. And so the summary here is that in addition to all the molecules in the nucleus cytoplasm of the infected cells, IFS16 recognizes the viral genome in the nucleus, and that is transported to the cytoplasm and to activate the pro-caspase activity. And also, our group and others have shown that it also comes to the cytoplasm, interact with the sting to induce the interferon response. So, however, we didn't know that exactly what is the mechanism by which the virus is IFS16 recognition leads to cytoplasmic transport, and how that it is what other proteins are involved in this one. So we just recently published that we found that the nuclear protein BRCA1 is interact with the IFS16 that mediate the nuclear innate sensing of the viral DNA, both the KSHV, EBV, and simplex 1, and also in the subsequent induction of inflammasome and interferon response. The mechanism by which the IFS16 is transported is because after the IFS16 recognizes the viral genome in the cytoplasm, in the nucleus, it gets acetylated. That acetylation leads to interaction with the ASC or to the transport to the cytoplasm to interact with the sting, activation of the interferon molecules. So in addition to the regulation of the inflammasome activation, we found that if you eliminate the IFS16, the virus simplex 1 replicates much faster, and the gene regulation goes up unchecked, very high level. And if you indeed knock down the BRCA1, the virus is produced much more in higher quantities. This is in simplex 1. Then what is the basically bottom line of the studies are, in the absence of IFS16, the promoters of the simplex 1 response, the immediate early or late genes, are all occupied by the viral transcription factor. They can activate the viral genome, whereas in the presence of the IFS16, these promoter levels interactions of the RNA pol and TBB is highly reduced because of the chromatin changes induced by IFS16. This is not only for the simplex 1. There is a paper also published with my collaborator in human cytomegalovirus also. If you knock down the IFS16, the virus replicates much higher levels. And this is also the interaction with the human papillomaviruses. This is with my collaborator we just recently published. If you take the cells which do not have IFS16, the viral DNA replicates much faster and at a different time point. But if you have IFS16, the viral DNA replication is restricted. So the summary slide is basically IFS16 is a cell cycle regulator, which is in the nucleus. It is also a transcription modulator. It's a sensor of a foreign herpes viral genome. It does not recognize the integrated lentiviral genome. The recognition can lead to inflammasome, it leads to interferon response, and also it can have a viral genome regulation. Why this is very important is, so that all of you know about the gene therapy stuff, the wild expression that those genome has to go into the nucleus. IFS16 will suppress anything that you put in to get it into the nucleus. Not only in interferon response, inflammation can be induced, but it will suppress the gene expression from there. So it has a lot of implication related to the gene therapy thing. So you have to overcome this particular way the IFS16 regulates any foreign gene expression from episomal DNA. So this has a lot of work we are working on these directions, as well as several laboratories in the world. So essentially the bottom line is it is an IFS16, it's a nuclear innate sensor. And what we have found is the latent herpes viruses, EBV, KSHV, and other viruses actually uses the IFS16 for their advantage for maintaining and establishing latency. Whenever the virus wants to come out and replicate the genome replication, it have a mechanism to overcome that IFN-16 response. So this is my last slide. This is done with a lot of my postdoctoral work, laboratories, and also we have funding from NIH. I'll stop here and I'll take questions.
Thank you very much.